Neurodevelopment is the study of how the brain develops from its earliest stages in the embryo through birth, and even beyond as the brain continues to develop throughout childhood and adolescence and into adulthood. We study neurodevelopment for several reasons, not the least of which is that it's fascinating. The process through which a fertilized egg grows to become first a minute ball of undifferentiated cells, and then over the course of weeks to months further differentiates to give rise to all of the different tissues in the body, including the brain, that's fascinating. Moreover, when we understand the steps that underlie neural development, it gives us insight into circuitry, into brain anatomy, and into function. For clinicians, understanding neurodevelopment is important because many of the disorders that we see arise during development. In addition, the same cellular mechanisms that are used to first set up the brain during embryonic development are often reused as the brain continues to grow and take on more complicated tasks. It provides a model for plasticity in adult life, whether we're looking at the processes that allow the brain of a newborn baby to grow and take on the much more complicated cognitive and motor tasks of an adult, or if we're looking to see the changes that occur in the brain between a young adult and one of an older individual. Finally, these mechanisms may be relevant for regeneration after injury. For example, developing new therapies to treat and repair damage after an insult, such as a stroke, may involve reactivating some processes that occurred during development. Now, because brain development is very complicated and because different people are interested in it for different reasons, this is a pretty huge topic. Adding to the complexity is that what we know about brain development comes from a whole bunch of different model systems. For example, some of the earliest steps in development that underlie the initial cleavages come from studies on sea urchins. Their external fertilization and the fact that you can get lots of sperm and lots of eggs from these animals has been important as a model in development. Historically, development is also widely studied using a model in chickens. The advantage here is that chickens lay eggs, and those eggs can then be windowed and opened up, and that allowed early experimenters to follow the normal development of the chicken embryo, including its nervous system, as well as to do manipulations by applying different kinds of chemical agents, and even doing things like transplanting limbs from one organism to another, and taking cells from quails and putting them into chickens. When it comes to genetics and identifying the genes and molecules involved in development, it's hard to overstate the importance of the fruit fly Drosophila as a model system. Many of the genes that we've identified in fruit flies also mediate the development of the brain and other tissues in mammals. If a vertebrate system is needed, another model system is a zebrafish whose clear embryos have made it possible to study development over a short period of time. For studies requiring a mammalian system, the mouse is a preferred model organism. In fact, it's possible to make transgenic mice and introduce genes from humans and follow their development then in that mouse system. And finally, we study development in humans themselves, whether it's from autopsy specimens that have allowed us to visualize the fine details of brain development, or more recently, from using MRI studies to visualize the fine structure and sometimes the functional development of connections within the brain. Gaining a full understanding of neurodevelopment is a huge task. For now, I'll simply emphasize that it's a multi-step process. It begins when the embryo sets aside cells that will become the nervous system. Next, those cells have to differentiate to regionalize the brain, so that different parts of the embryonic brain can develop into different regions. The front part of the brain will develop into the cerebrum. A middle part of the brain might develop into the hypothalamus, and parts from the posterior part of the nervous system will develop into the spinal cord. Another key step is generating all of the neurons and glial cells that will make up the brain. We have about 86 billion neurons in the human brain, and almost all of those are born during embryonic development. We'll see that those neurons are born deep in the brain, and then they need to migrate out in order to find their final locations. Once there, the neurons will differentiate so that some neurons will take on the appearance of Purkinje cells, whereas other neurons might take on the appearance of a pyramidal neuron, and still others will look like stellate cells. Curiously, we'll see that the nervous system makes more cells than it actually will need, and there's a process of programmed cell death where excess neurons are eliminated during neurodevelopment. Another important step is the outgrowth of axons. Neurons might have their cell body in one part of the brain but need to send projections out that travel tens of centimeters in order to reach their target tissues. This process of axon guidance is important for establishing functional connections. Once the axon reaches the appropriate target, it needs to form synapses. This process involves competition. There'll be even more cell death. The synapses, once formed, show some plasticity. There's often synaptic rearrangement and fine-tuning that occurs. The crude initial pattern of synapse formation later gets refined by rearrangements based on experience and feedback. And once the pathways and the circuits are established, the brain will continue to develop. One of the important things that will happen is there'll be increased myelination over the course of development. These many steps here are incredibly complicated, but also very interesting. And in future videos, what I'll try to do is break them down to walk you through these, to talk about the mechanisms that underlie these different steps.